I'd like to welcome you to the presentation by Steve Giovannoni, a distinguished professor of microbiology. He's one of 2 Oregon State University faculty members to be awarded the title of Distinguished Professor this year. This is the highest honor that the university gives to a faculty for his teaching, research, and other outreach activities. For Steve, he is being honored for his teaching, collaborative research in marine microbes, and advanced molecular techniques. Professor Giovannoni's studies of oceanic Bacterioplankton have led to the discoveries of worldwide importance, strongly influencing the field of microbial ecology. He originated the identification of uncultivated bacteria by amplification of rRNA genes, discovered previously unknown dominant taxa information and world geochemical cycles, and he continuously pioneers techniques for microbial ecology research. Professor Giovannoni, who received his doctoral degree in biology at Oregon State University, has been with us since 1988. His work on marine bacterioplankton from the oceans, seafloor rocks in Antarctica is at the forefront of microbial molecular ecology. He discovered SAR11, now known to be one of the most abundant microbes on the planet and a major player in the ocean carbon cycle. I know he's going to be talking about some of his research, so I don't want to steal too much of it. But he has been a pioneer in this particular field. Steve has been awarded over $8 million over the past 10 years, including a $6.5 million Gordon and Betty Moore Foundation grant. He's been awarded the Pernot Distinguished Professorship in Microbiology at OSU and the Milton Harris Award for Exceptional Achievement in Microbiology, and named Fellow by the American Academy of Microbiology. It is my distinct pleasure to welcome Steve Giovannoni, the 2012 Distinguished Professor of Microbiology.
Thanks, Giovanni, and thanks to all of you for coming. Can you hear me all right? And this is a little bit like teaching a class in that there's a broad selection of colleagues here, and when you teach a class, you have to kind of aim for the middle. So I hope there'll be something in this lecture that will be understood by everybody here. It's going to go into some detail, but I have a big lab, so I will touch on quite a few topics. So let's get started. So this is where the talk starts. This is an electron micrograph of Bacterioplankton, by the way, taken with the new EMs that have been installed over in the Linus Pauling Research Center. And microbial ecosystems everywhere are populated with organisms like this, which are interesting to us because they catalyze chemical reactions, particularly biogeochemical cycles. When I got here to OSU in the late 80s and throughout the early 1990s, my lab and other labs were engaged in research to try to identify these organisms. And the single most important conclusion that came out of all that research, not just ours, but everybody's, was that most of these organisms, by the way, is there a pointer? Nope, okay, I'll use my hands. Most of these organisms can't be cultured or couldn't be cultured easily in laboratories. So there are lots of bacteria being investigated in laboratories around the world, but it was understood as a result of these applications of molecular technology to microbial ecology that really a lot of the important things in nature probably were not cultured. And so that's how we got started on the work I'm going to be talking about today. But next I want to tell you why it's important. Oh, there we go. Top button.
All right.
All right. Oh. So this is a diagram of the carbon cycle as it occurs in the oceans. It's very simple. We have CO2 being converted to organic carbon, and that organic carbon being converted back into CO2. Some fraction of that material is leaving the carbon cycle and becoming sequestered in some form. And that's why most of the research in my laboratory gets funded. That's one of the major motivations for funding it, is to understand this process. To put this on sorry, I'm a little mixed up here. The system back right here. These diagrams on the right-hand side of the slide show you distributions of dissolved organic carbon in the oceans. I like to describe this, think of dilute chicken soup out there globally. This work done by Hansel and Carlson. Carlson, a close collaborator with my laboratory. And the important point about this is if you integrate this carbon globally over the entire surface of the planet and sum it all up, the total amount of carbon stored as dissolved organic matter in the oceans is about equal to the total amount of carbon dioxide in the atmosphere of the planet. This is the largest pool of reactive carbon on the planet, and interactions between organisms and this carbon have the potential to dramatically affect the amount of CO2 in the atmosphere. Fortunately for us, there's a process known as the microbial carbon pump, which is somewhat mysterious, in which this carbon is transformed into molecules that are resistant to oxidation, and therefore it accumulates, as we see on the right-hand side of the graphic. The average age of this carbon is about 6,000 years. So this so-called microbial carbon pump has a dramatic influence on the atmosphere of the planet. I talked about that in a lecture I gave, and more broadly in a lecture I gave about a week ago. And it's motivations like this that cause us to, want to understand microbial life and biochemistry. So I decided to pitch this talk to discuss in this talk the High-Throughput Culturing Lab. This is the name, it's an unofficial name. The HPCL is basically my laboratory. And I want to give you a little history and tell you how it got started. The HPCL was founded in the late 90s when we decided to tackle this problem of why are these organisms difficult to culture. So everybody at that point had accepted that they were called, in fact, unculturable by many laboratories at that time. And everybody was taking the tactic that they would attempt to understand these entirely with genetic approaches. We felt that taking more, I won't call them traditional, but taking approaches that involved growing the organisms would be important to understanding their geochemistry. So we set out to grow them, and we did it by creating a series of hypotheses that we thought could explain why they were so difficult to grow. And so one of those hypotheses was that slow growth and low cell densities might not be detected using conventional methods in use in laboratories. Another is that these organisms, you might not be able to isolate them simply because they're interacting with other organisms, and if you take away the interaction, they won't grow. Another is that they might simply require very specialized growth conditions that we could only guess at, and without guessing correctly, they wouldn't grow. And a fourth possibility is simply contamination in our laboratory could inhibit their growth and prevent us from growing them. So we created the new technology that would address these hypotheses. And I'm going to go through this pretty quickly, but we decided that we would try to isolate these by extinction culturing, which means diluting cells until there are so few that there's only one in a chamber, and allowing it to grow. To do this, we wanted to miniaturize the process. We wanted to be able to detect less than 1,000 cells because we calculated that if we were in very small volumes in microtiter dishes with the kind of technology that can be scaled up to high throughput, that would be the most effective approach. And we needed identification methods. So it doesn't do any good to grow an organism if there are thousands of them and you can't figure out which one is interesting and to focus on. And finally, we used robotics to speed things up. So this slide shows an array of equipment that we purchased for this, and this story is a good one, a good story about OSU, because when I got this idea, I knew I needed resources to pursue it, and those resources came from a series of grants, the major research instrumentation grant from NSF, a grant from Murdoch Charitable Trust, and matching funds from many organizations at OSU, including the College of Science, the Research Office, the College of Agricultural Science. Everybody stepped forward and put some money into the pot to support this research. I have a funny story, I don't think I have time to tell, about how narrowly it was funded. There was a lot of criticism of the project, that it was too ambitious and that the ideas were untested. NSF likes ideas that they feel secure will produce an outcome. And there was a lot of concern about this proposal, but in the end, everybody pitched in, and we were able to get the funds and pursue it. And and sorry that I'm off my my rhythm a little bit here this morning. And about that time, a film was made about an organism we've been working on in the laboratory, SAR11. So I thought I'd show it to you. So this movie was made by NSF and the microbiological, the ASM, and it was shown on TV. And if you've ever had anything done like this about you, it took me months, maybe a year, before I could actually watch it. And when I watched it, I discovered that the cartoonist who was involved in this really had a sense of humor. So here's his depiction of SAR11, one of the organisms that we were after. And he's running, and he sees something, and he's running off screen. And what he sees is me, because I had a big handlebar mustache then. And he sees me after him, and so he runs for it. So I realized this is pretty funny, because that's exactly what was going on at the time. We were pursuing this organism, hoping to isolate it. And this was the technology that we used. It's pretty simple to understand. We started with seawater. We sterilized one flask, distributed it in a microtiter dish, diluted these cells until a known number of cells were in each well, and then incubated them, let them grow, and then screened them using a screening technology we developed, a cell arraying technology we developed for this purpose, and then looked to see what we got. And by the way, the most important ingredient turned out not to be the robots, but Stephanie Kahn and a good graduate student who diligently pursued this project. And there's no question that this has been a very successful approach. So we've published many papers describing new genera, families, and species, and I think the last new phylum described on Earth was by us, this one, Lymphosphaera, by a postdoc, John Cho. And so it's been very successful, and another part of that success is illustrated in this slide. It's hard to describe to you how this work has influenced laboratories around the world. We have thousands of cultures. Most of them won't grow on petri dishes. I think only 8 of them are with the HPC National Culture Collection at this point, but more are on the way. We sent many of these strains, this says 40, but I think now it's up to quite a bit higher number, to researchers, extracts. We've sequenced 25 genomes and deposited them in GenBank. There's a patent about this work. And really I'm just going to skip ahead and say a lot of the discoveries that are made based on what we've done are made by people who go to databases, see things in the genomes that are interesting, contact us for DNA, or else have the gene synthesized, or they may want the cells. And one example right here at OSU is the discovery of xylose isomerases, which I think Trillium Fiber is now working on. So this biodiversity that is captured by our culturing approach and other scientists doing similar work is a real resource for research around the world. So I'm going to tell you a little bit more in the next section to talk about bacterioplankton. This is, of course, a map of the globe showing chlorophyll concentrations. We work at a site, Bermuda, indicated by this little arrow. It's not down in the Caribbean, it's way out in the Atlantic. And we go there because it gives us an opportunity to sample open ocean regions. We have a lot of research going on there, but I want to just tell you a few things about what it's like in the open ocean and what the ecosystem these bacterioplankton live in looks like. It's an ecosystem where there are periodic fluctuations, so this is many years of data. This is nitrate and phosphate here. This is total dissolved organic carbon, which you see fluctuating. The dynamics of this fluctuation are something that's of intense interest to oceanographers studying the carbon cycle. This is the composition of that dissolved organic carbon. It happens to be dissolved neutral sugars here. And this is the total abundance up in this corner of bacteria in the water column. I didn't mention it, but the numbers are about 500,000 per mil. That's true worldwide. So these are the organisms we're interested in, and as a result of genetic data, the accumulation of genetic information about them, we can now tell you things about their ecology even though we can't grow them. So some live in the surface and bloom in the summertime. Some live in what we call a mesopelagic, below the euphotic zone. Others have more complex, this one have more complicated distributions in the environment. So this ecosystem is not homogeneous. It's a very structured ecosystem. This is the result of a lot of work from my lab and others. And because of having genetic markers for these organisms, we can go out and map this kind of diversity, or this kind of ecology, without understanding what the organisms actually do. So we're giving them a name, and we're looking at the patterns in their occurrence in the environment without knowing what they do. So that's where culturing came in. And we were fortunate that we were able to culture our #1 target, which was SAR11, a little alpha proteobacterium. And this is Bob Morris, a graduate student who published a paper in Nature saying that there were about 24% of all the prokaryotes in the ocean. And today, most microbial ecologists would agree that the best data indicates that these are the most abundant organisms on the planet. Now, they're little teeny bacterial cells. One of the smallest cells known, in fact. These are cryo-electron tomography images of them. These little dots you see in here are ribosomes. And the orange shading here in this image is the nucleosome of the cell, so the DNA of the cell, which takes up about half of the cytoplasm. But the most interesting thing about these organisms we learned when we sequenced their genome, which is that they had, at the time, the smallest genome that had ever been seen in a free-living cell. So in the next part of the talk, I'm going to tell you a little bit about this. It might get a little technical for you non-genomic scientists, but it's been an important part of our research, and it's had a pretty big impact. So this chart shows number of genes on this axis and genome size on this axis, and it's almost a linear relationship because in bacteria, genes are all, have an average size, and most of the genome encodes for protein-coding genes. So everything that's in green here is a free-living bacterium. If it's a parasite like a parasitic disease-causing bacterium, it's a little blue circle. If it's a typical marine bacterium, it's a little yellow circle. And Pelagibacter is down here in red, and all the black symbols on this side of the side are obligate symbionts, including pathogens like rickettsias, chlamydia, and mycoplasmas. So it's right there at the boundary between those things with bigger genomes, which are presumed to be more complicated, and the very small cells that are involved in symbiotic relationships. Now, there's theory that describes why cells might have small genomes, and it's been fairly controversial theory, but I'm going to mention it. One of the most important papers was published by Lynch and Connery in 2003, a paper titled The Origin of Genome Complexity. And by the way, John Connery was my college roommate, and he's down at the U of O, and John happened, by the way, I hate to admit this to Sabah, but I'm actually a duck. I got my PhD at U of O. And what this graph shows is the effective population size times the nucleotide mutation rate for a lot of organisms, starting with, there's a mouse at this end. human Homo sapiens is right above the mouse. And then at the very far end is a Prochlorococcus, a very abundant marine bacterium. So you can see that these numbers go up as we come up this graph. And to a non-evolutionary biologist, let me put it to you this way. Genetic diversity is created by a number of processes, but one of them is mutation rate, and it's controlled by effective population size. So a cell that goes through truncations in population size, this would include a lot of pathogens, but anything with a small population size, a lot of diversity created by mutation is going to be eliminated. But in an organism that reaches very large population size, that diversity is allowed to persist. We call that elimination of diversity genetic drift. It's a very well-known factor to population geneticists. And essentially what Lynch and Connery were saying is that, well, organisms like humans and mice have very small population sizes, and so there's really a lot of genetic drift. But selection, natural selection that perfects biological machines is very efficient in organisms that have very, very large population sizes. According to this theory, it's possible for selection to eliminate DNA with no real function or a minimal positive fitness from very large populations of organisms. And conversely, organisms that have very small population sizes, the opposite happens. Random drift can cause the loss of genetic information. And so here's an illustration from a paper by Moran and Achman showing the relationship between mutations that cause a substitution in amino acids to those mutations which are silent and cause no change in the protein encoded, and the log of genome size. And you can see that as we drop down in genome size, we have a tendency for more and more of the mutations to be random mutations in the genome. In other words, a consequence of genetic drift. So what about this organism I'm telling you about, SAR11? Here's some data from that organism. Here, all this data supports the idea that Lynch and Connery's hypothesis applies to these organisms, that selection is acting very actively, eliminating DNA with no useful function from the cells, very small amount of DNA that does not code for proteins, very little expansion of genome size by gene duplication, as indicated here, a tendency for the cell to have a higher proportion of amino acids that are low in nitrogen content, such as lysine being substituted for arginine in the proteome of the cell. But despite all this evidence for a lot of selection for minimization of the genome, there was also evidence that the metabolism of the cell was relatively complete. So in these cells, like rickettsias, that are parasites that have small genomes, the loss of genome size is often associated with the loss of ability to make growth factors that the cell needs. For instance, amino acids. If you're a parasite of a eukaryotic cell, you don't need to make tryptophan because you can get it from your host cell. Now, there were alternative explanations for the small genome size in Pelagibacter, and one of those alternative explanations might be, well, perhaps they're just cut off from DNA from the external environment. Most bacteria exchange DNA very rapidly with other organisms in a process called horizontal gene transfer. And so one potential argument for the small size of this genome is that these organisms might lack those kinds of genetic mechanisms. But Kevin Vergeen, who's over there in the audience, published this paper showing in fact that Pelagibacter, which is a synonym for SAR11 in this talk, has the second highest recombination rate ever recorded for a microbial population, meaning that Pelagibacter cells in the water column coexisting are exchanging DNA at a very high rate. So they obviously have mechanisms for the exchange of DNA and mechanisms for incorporating that DNA by recombination into their own chromosome. And what's more, when the genomes of this organism were investigated, this is a genome here by one of my graduate students, Wilhelm, the genome shown from left to right, gene by gene. It was discovered that they have hypervariable regions. These hypervariable regions were confirmed when we got more genomes from additional strains of SAR11. And here we see a figure from a paper that's in review right at the moment, showing that there are in these genomes hypervariable regions in which the cell is inserting DNA from foreign sources, so it has currently a very active process of horizontal transfer going on. We also know, by the way, that this organism is susceptible to phage attack. So here's a cell that's been populated by phages. There were rumors that they might be immune to phages, but that's not true. Jan Zhao, who's out there in the audience, has isolated the number of phages of this cell. And it's believed that these phages are one of the vectors that brings foreign DNA into the cell. Finally, this is a figure from that same paper that's in review in collaboration with Microphase Lab in Hawaii, a graduate student and postdoc, in which 7 genomes from SAR11 strains were compared. And what we observed in those genomes was that they all had very small genome size. So it's not as if the one isolate that we're working with happens to be an anomaly. They all have very small genomes. They have the highest conservation of gene content ever observed in bacteria as a percentage of the total genome size. And so we think that the observations that we've made are perhaps not universal for all SAR11s, but apply to many organisms in the clade. So let me summarize this part of the talk by saying that I've told you about 2 different pathways by which cells can get very small genome sizes. And one of those pathways, seen in symbionts, we have low population sizes, genetic drift, and a reduction of the core metabolic repertoire, increases in pseudogenes, and lots of gene duplications. And the other pathway, which we see in SAR11, we have very high effective population sizes, very strong selection. Just over 1,000 genes is the size of most of the genomes we've encountered in highly streamlined organisms. There's a stable core metabolic repertoire. There are occasional pseudogenes, but they're very rare. And intergenic spacers are minimized, and apparently as a consequence of streamlined selection. So here's the most successful organism on the planet, as far as we know. And it turns out that it's an exceptional organism in terms of its population genetics and its genome architecture. It's one of the smallest, most conserved genomes out there. So how do you reconcile these observations, this incredible simplicity in the structure of this cell with its tremendous success in an ecosystem where they, by the way, if you see them in the water column, they're bobbing around freely by themselves. How do we reconcile these facts? So the next part of the talk, we're going to examine what some of the consequences of genome streamlining are for how these sort of cells are organized, both in terms of their regulation and biochemistry. One feature of these cells that we investigated was their what were structures known as riboswitches. A riboswitch is a region of a messenger RNA located upstream of the region of the message that codes for a protein. And what this part of the RNA does is to fold up into structures shown here in this diagram. And those folded structures have the ability to interact with small molecules in the environment and regulate whether or not that transcript is converted into a protein, so whether or not it's translated. So there are more riboswitches known in SAR11 per gene, per amount of coding DNA than any other organism, and more new organisms were discovered, more new riboswitches were discovered in this organism than had been known previously. Why was it such a rich topic of subject for research into riboswitch diversity? And the answer is because there's so much genetic information available about this organism as a consequence of metagenomics. Comparative study of that information made it possible to identify these folded structures and to investigate their functions. And we've published a series of papers about this. I'll say a little bit in the talk, but I want to mention the student who did a lot of this work, Daniel Smith, up here, shown in the corner, who's recently left for a position at Argonne National Labs. And one of the things Daniel is reporting in a series of papers from his PhD thesis is that these riboswitches, acting in conjunction with other mechanisms in the cell, essentially had decoupled in this cell transcription from translation. So in this figure, you see the correlation of messenger RNA change with protein change. This is quantitative proteomics being compared to microarray data. And this would be a correlation of -1 and a correlation of +1 and 0. And overall, the correlation between the expression of a gene, the transcription of a gene, and its translation in this organism is 0. And a lot of that has to do with uncoupling. So it has a very simple regulatory system with far fewer regulatory elements than we're used to seeing in bacteria. And a much higher reliance on riboswitches to control expression. So I'm going to shift now to talking about some of the metabolic consequences of genome streamlining. One metabolic consequence was described in a paper in Nature by Jim Tripp. Here's Jim, who's now down at the Joint Genome Institute. And Jim observed that these organisms were deficient in many of the enzymes that are involved in the assimilation of sulfate into sulfur in proteins. So this is a function that's almost universally present in aerobic bacteria, but appeared to be absent in SAR11, and his experiments confirmed that that was the case. So this is one of the first examples we had of a nutritional deficiency created by genome streamlining. So something has to be missing to make a genome this small. And one of the first things we found that was missing was this ability simply to use sulfate as a source of sulfur. And Mike Schwalbach, shown here in the corner, showed that oftentimes these organisms are missing even glycolysis, which is one of the fundamental pathways in metabolism. And there's a relationship between strains from the coasts and those and the glycolysis operon. So as chlorophyll increases in concentration, it becomes more likely that isolates will possess this operon for putative operon for glucose oxidation. We don't know the reason for this yet, but it appears that streamlining is so hammering these cells very hard out in central low productivity regions of the ocean and causing even this important part of metabolism to be deleted. And here's some work taken from a couple of papers, one published and one that's now in review by a graduate student named Paul Carini, who's out there in the audience, which shows some of the unusual organization we've observed in metabolism for this cell. This one happens to involve a riboswitch. There's a riboswitch that normally controls, in many bacteria, the degradation of glycine to CO2 and ammonium. In this organism, there's a duplication of that riboswitch. It has a different Km for its substrate, and it appears to regulate a reorganized version of intermediary metabolism in which these cells are taking glycolate that probably comes from phytoplankton in the water column, and that glycolate can be converted into, can be either oxidized by conversion to malate, or it can be converted into glycine. And they're using these riboswitches to regulate this pathway so that when they run low on glycine, they're not oxidizing glycolate, one of the glycine, which is a potential precursor for glycine formation. Now, another surprise that came out of reading this genome was the metabolism shown in this slide. Another graduate student, Jing Sun, out there in the audience. We were very surprised to discover a large number of genes in this organism for the oxidation of very low molecular weight compounds, including methanol, formaldehyde, trimethylamine oxide, trimethylamine, and other so-called one-carbon compounds. So Jean came to the laboratory a couple of years ago and published her first paper in PLOS One last, I think it was almost 2 years ago now, describing this unusual metabolism in this cell. We're still trying to understand the implications of this. So here's an organism which has a very apparently limited metabolic repertoire, and yet one of the things it's choosing to focus on are these very low molecular weight compounds in the environment. So these compounds were not thought by oceanographers to be an important part of the carbon cycle, and now that we realize that that these cells are targeting these compounds for oxidation, we've turned around and gone out into the environment. So my group does go to sea on ships and do experiments on shipboard, and here's the result of one of those shipboard experiments, showing that the kinds of results that Jing observed in the laboratory could be reproduced at sea, and that these natural populations of cells have a very high capacity for the oxidation of these compounds. We're trying to understand now why apparently so much of this of compounds in this class is present in the water. This is a diagram of SAR11 metabolism. It summarizes what I told you. So I started out saying that we wanted to understand how they interact with dissolved organic carbon. So that's why so much biochemistry is being done in the laboratory. What we can tell you right now is that they tend to shy away from carbohydrates. They like organic acids, amino acids, polyamines. They like C1 compounds, and they like a group of compounds known as osmolytes, which are often methylated, and this would include dimethylsulfonyl propionate, and taurine, glycine betaine. These are all osmolytes that SAR11 cells appear to prefer. DMSP is actually particularly important to oceanographers and climate scientists because its breakdown product DMS is involved in the precipitation of cloud formation. Now, collectively, all this that I've described to you is a very different view of metabolism than what you would normally get by going into a microbiology textbook and reading about how bacteria work. And so I'm going to give you a couple of models that will help you understand that. I like to call the model that is most, really the paradigm in microbiology is the alternate substrate model, in which a bacterium, we put it on a petri dish, we can give it glucose or we can give it tryptophan as its sole carbon source, and when we change the carbon we give it, it simply switches from one to the other, and it's able to grow as long as it has all the elements that it needs for growth in the right forms. And so I call that the alternate substrate model. What we're seeing in SAR11 is something, a very different model that I would call a balanced substrate model, in which the networks of metabolism have been greatly reduced in terms of the number of connections, resulting in a change in really how the cell functions. So if I would use the analogy that the metabolism of a typical bacterial cell is something like the vein of a leaf. If you cut one vein in a leaf and put that leaf into fluorescent water with a fluorescent compound in it and shine a light on it, all the veins of the leaf will be filled with the fluorescent dye. And that's because the design of a leaf vein network design is there to overcome damage that might be created by insects. But if you go out to the wrong end of a subway line and discover that it's 2 in the morning and you're at the wrong place, oftentimes to get to your destination, you've got to go all the way back to the hub and then back again out, because there are far fewer connections. So systems engineers call these distributed and decentralized networks. And our hypothesis is that what's happened over at streamlining the course of streamlining selection is that many unnecessary genes have been deleted from SAR11, leaving it with a metabolism that's very effective, but also makes it much more dependent on obtaining a balanced supply of substrates of different types for it to grow well. Now I'm getting near the end of the talk, and I have a couple more, I think, important points that are important to me to make and are relatively general. Well, one is a short story about why it's important to have cells in order to test hypotheses that emerge from genomes. So a lot of the change in the field of microbial ecology over the last decade has been an emphasis on avoiding culturing, so by obtaining genomes out of nature, which gets you around the problem of, it gets you around a lot of work, and it gets you around all the challenges of cultivation. But that approach is based on the assumption that you can reconstruct metabolism from gene sequences. And in this organism, if anything, over the last decade of our work, what we've discovered is we would have a very inadequate idea of the metabolism of this organism if we were trying to infer it all from genomes and didn't have the ability to do experiments. So here's just one example. The simplest system in the cell also attracted some of the greatest interest from oceanographers. It's a light-dependent proton pump called proteorhodopsin that in the light simply pushes protons out so that the cell can make ATP without performing respiration. So this was the first organism isolated in culture which actually had this protein. So it gave us a chance to test the hypotheses that were out in the literature at that time. And this is a curve, sorry, some parts of the graphic have disappeared, showing growth in the light and in the dark for SAR11, and that doesn't matter which symbol is which because the curves are nearly identical. So light's having almost no influence on the growth of the cell. And we did many experiments like this, and we couldn't figure out why, because we knew they made the protein, and we knew the protein was active. And finally, Laura Stindler in my lab solved the problem when we started measuring oxygen consumption. And it turns out that this one simple protein is integrated metabolically such that it's present all the time, but it's inactive unless those cells are starved for carbon. So these cells do the same thing that we do when you starve us for carbon. We shrink. We continue to respire, but we respire endogenous reserves, and our biomass gets converted back into CO2. The same thing happens in these organisms. If you shine the light on them when they're starving for carbon, you'll discover that the oxygen uptake rate drops dramatically in the light. So respiration is slowed down, and the cell simply turns to this source of sunlight as power under those conditions. Now, to figure out that that's how this one single protein operated at a physiological level took us years of experimental work. And I think it's a good argument for why we need to have cultures and we need to study them in order to understand what these cells are doing in the ocean. I've got, and this is a picture of starved cells that are shrinking, and starved cells in the light that look nice and happy because they're getting power from the sunlight. Now, I want to widen out. I've spent all this time, really, a lot of this time dwelling on one very important example of a single organism we got using the HPCL's approach to culturing. But there are some others. One that was isolated recently called OM43 here. and another called Prochlorococcus, which researchers around the world and MIT work on. So there are a few organisms like this that have very streamlined genomes and are difficult to grow. I wanted to mention this one, which is common off of our own coast. This is a paper recently published in collaboration with Kim Halsey, who's shown here. So this actually has a smaller genome than SAR11. It's a very difficult organism to grow. It's called OM43 or strain 2181. And its a unique characteristic is that it's an obligate methylotroph. So it's further evidence for us that methylotrophy, or the oxidation of C1 compounds, is an important part of the carbon cycle that had been overlooked. So one point to showing this is that there are lots of organisms in our cultured collection which we're currently not studying, and that we're hoping other investigators around the world will jump on those organisms and to try to understand what's going on with them. I'm sorry I can't tell you about, I know that you're disappointed too, that I can't go through the biochemistry of this very interesting organism right now, because I would love to. But I couldn't do as good a job as Kim, so I better keep my mouth shut. But I do want to say that this raises a question. There are lots of genomes of marine bacteria that are typical size for bacteria, and now only a few genomes that have been isolated, some from, albeit important, organisms. So why is it that there are so few of these in culture? So one of our hypotheses, which is a testable hypothesis that I expect to see an answer to soon, is it may be that many of these organisms out there in nature are difficult to grow because they also have streamlined genomes, and that streamlined genomes in general tend to confer organisms with the property of having complicated nutritional requirements, in the sense that a complicated balance of the right substrates for growth. So if that's true, we're going to find out soon, because single-cell genomics is now coming of age, and it's becoming possible to take cells out of nature and amplify their genomes without growing them. And while that might not tell us everything about their physiology, it will certainly tell us whether or not they have streamlined genomes. And we'll answer this question: is this an explanation for why so much of diversity is uncultured? There's another way to think about this. Ocean scientists studying broader ecological processes have observed a very high level of connectivity in these populations. And I know I've gone over a lot of topics, and I haven't gone into the details much with you. But think of it this way: we're used to looking out the window and seeing organisms in our ecosystem interact. But these bacteria in the ocean, all they are is dots under a microscope. So the evidence for them interacting is statistical evidence from long-term studies of their distributions in nature. And some of these distributions are shown here in these plots, often unique for different species. So the question becomes, are these plots of correlations in the distributions of organisms the consequence of nutritional ties? In other words, has streamlining selection reduced the size of these genomes to the point where organisms have become dependent on compounds made by other organisms, and that first makes them difficult to isolate practically for us, and secondly, ties them much more tightly into ecosystem networks. So that's also a testable hypothesis. We don't have an answer yet, but I expect to see an answer to this question coming out from various laboratories in the next few years. Finally, this is partly something I haven't talked about much, but I made this slide, these last two slides for this talk. This is a plot of several kinds of information from the ocean at the Bermuda Atlantic Time Series study site, that site that you go to in Bermuda. And this is 0 meters and 300 meters. And this is RNA DNA ratios, which you see dropping dramatically as we drop down through the water column. Photosynthesis is indicated by the green dots. And the RNA, let's see, and what are the blue dots? And blue dots are species richness, not really relevant to this talk. So why do RNA DNA ratios change so dramatically as we move down through the water column? Well, for one thing, this microbial community in the euphotic zone and this one in the dark region just below are completely different groups of organisms. Amazingly, there's a tremendous change in the structure of the community, and not just the photosynthetic organisms. The other organisms that recycle carbon also are changing. And RNA ratios are dropping dramatically, probably because there's so much less energy flowing through this part of the system. The energy is all up here where light is supporting the ecosystem through photosynthesis. So these organisms that live down in this lower part of the water column are some of the toughest organisms to isolate. We have not been successful at the HPCL in getting them. We've gotten a lot more of organisms from the surface and very few from down here. So I'm going to throw a different hypothesis out for why they might be so hard to grow. It could be simply very low growth rates down in this part of the water column. Up here, the cells are dividing at a blazing rate of about once a day. Now, most of my microbiology colleagues think that that's a very disappointing growth rate. But it's actually, if you think of all the cells in the oceans, which have more biomass than all the whales and fish, all the bacterial cells have more biomass than all the whales and fish put together. You can almost hear the thump if they all divide once a day, right, globally. That's a huge amount of biomass being created. But down here, we probably have a much lower growth rate. So this morning, I did the calculation. And it's kind of for my students here. So if you set experiments up the way we do in the HPCL, we take one cell, dilute it into a well, and we wait 3 weeks and we assay that well to see if they've grown. Now, if a cell divides once a day, how many cells do you have after 3 weeks in that microtiter dish well? Everybody? Probably not many of us academics can actually do that in our heads. I once met a train conductor who could do that and a lot more. He was very fond of dazzling college students with this ability. Well, the answer is about 2 million. Did I hear 2 million out there? Did I hear that? Okay, now if that cell divides every 5 days instead of once a day, how many cells are going to be in that well at the end of 3 weeks? The answer is 18. Okay, so you see the problem. Large parts of microbial diversity may be beyond our ability to study simply because of our patience isn't great enough. But also because our technology isn't good enough. So we were successful because we pushed our detection sensitivities down by almost 1,000-fold. And it gave us access to a tremendous amount of microbial diversity. So we have thought of ways to address this problem, and we're working on it right now. So I see Zach there, so this is almost my last slide. So what we've done, this is Zach very recently. Zach has built a Microfluidic Workstation, and we're collaborating with Steve Quake's lab at Stanford. So we now have the ability to do single-cell genomics at a microscopic level. This is very exciting technology. I would love to show you more about how it works, but basically you find the cell you want and you drag them into a chamber and amplify its genome. So you do this all by manipulating the cell under a microscope. This microfluidic chip will do 48 genome amplifications simultaneously. So we're hoping that that this technology will drop our sensitivity for working with cultures down enough so that we can begin to work with these cells that are growing at which we suspect, one hypothesis is that they're growing at much slower growth rates, and hopefully get access to parts of microbial diversity we haven't been able to study previously. By the way, Zach, yesterday, the optical trap on the microscope worked for the first time, because Zach's been building this thing for the last year and a half. So he was pretty excited about that, and he's flying Zach and the entire instrument are going to Bermuda to hopefully amplify single-cell genomes. So obviously there are a lot of folks in the lab that contributed to this work, and I haven't been able to mention them all by name, but you can come over and visit us in Nash Hall and meet them all anytime you like. And they're on this slide. And I did thank my OSU for their support, but also outside support, of course, for this sort of work from the Moore Foundation, the Joint Genome Institute, and the National Science Foundation. Thanks for listening to me. Sorry I tripped on a few slides there.